Hello everyone. In this session we are going to study about the Human Genome Project. So in the last sessions we studied about the gene expression and the lac operon. Before that we also studied about the transcription, translation and everything. So today we are going to study about the Human Genome Project. What it is. So first of all we'll learn the concept name, what the word defines. So the genome is total constituent of the gene, the total content whatever present in a human body, the total number of gene together we can call it as genome. So we are going to calculate, we are going to identify and also we are going to find out the total genes, total DNA base sequences present in the human. Don't you think so? This is a very difficult one concept. How to identify the base sequences present in the human, overall in the human body. So we have number of cells, we have number of different type of cells. All those cells we have to isolate and clone and sequence in a particular manner, and how to read out, where to store, where to write all those things. This is quite bigger and this is quite difficult and this is quite complex concept or this project. But the US Department of Energy and National Institute of Health, they came out, they started this mega project in 1990. With them the UK Wellcome Trust, they also became a major partner for this project. What project it is? It is a Human Genome Project. What they are doing in this project, they are going to calculate, they are going to identify the total overall base sequence, how many base sequences are present, how many genes are present in the human body. We know no two individuals are identical. I am different. In me, the cells or the DNA this fingerprinting and the characters, everything is different. I won't match in a character or in any kind of mechanism with the one more fellow. Okay, so these differences, why these differences, how to understand why these differences came across. So these differences are nothing but having different sequences of bases in DNA. That sequence of bases in DNA makes up the genetic material. The genetic material we studied in the previous sessions, the genetic material or the gene is just a fragment of DNA. It means what? The gene it itself are having the base sequences. What base sequence? Nitrogen base sequences. How many nitrogen bases are present? 4 nitrogen bases are present. So adenine pairs with the thymine, guanine pairs with the cytosine, like this number of repeated sequences makes up the gene. So what they are doing, what the US Department of Energy and National Institute of Health, what they have planned, they are going to identify the total number of genes, the functions, how many base pairs are present in a human body. So when they started this project in 1990, the Human Genome Project, it consists number of concepts. First we are going to study about the aims of what? Human Genome Project. Okay, Human Genome Project. The first thing is aims and goals and methodologies and what is that? Salient features and applications. So these are all the things which is present in the Human Genome Project. The work started in the 1990. They estimated this project should complete in 13 years. It means when it should complete in the year of 2003, it should complete. What were their aims? How they planned to do this work? What is the what they planned? So this is how we have to work out on this project. This is all the things we have to find out. So those all comes under the aims. The aims were the estimation they did, the estimation they put. The estimations were like there will be estimation, the estimated base pairs present, estimated base pairs. Base pairs means how many base pairs are present in the human body. That estimation was 3 into 10 to the power of 9 base pair. So this was the estimation they put. In the aims, this concept, what they did, they estimated maybe this much of base pairs are present in the human body, and to find out one base pair, it cost $3. Estimation was like it cost to find out one base pair. It cost $3 per base, not to find out all the total base pair. To find out one base pair, the cost of to find out one base pair is $3. Next, the total estimated cost of this project is $9 billion. How much it costs? $9 billion. This is one aim, this is one aim, and this is one aim. So we have 3 aims now. So how many million dollars? $9 million. The total estimation of the US Department of Health and Institute, that I'll write here, National Institute of Health. Okay, and National Institute of Health, one more trust they joined with them. That trust is Wellcome Trust of UK. They became a major partner to this project. And then number of countries they came forward to help this project. Those countries are Japan and France and China. So these are the other countries who came forward to help this project. Okay, so this is the introductory part, and now we are in the aims of Human Genome Project. It cost $9 billion to sum up the total base pair. On that time, there was not much advanced technology to store, to retrieve, to analyze the base pair. Only one branch was there, the new arising branch of biology, that is bioinformatics. So because of having only one branch, the Institute of Health and Department of Energy, make it correct, Department of Energy, National Institute of Health it is. Okay, what they did, they started storing, started retrieving and analyzing the project, the genome project in this branch. And one more thing I must tell here, one branch they design. What branch it is? The branch is LC. So what is LC? LC means that this is the short form of ethical, legal, social issues. So this ethical, legal, social issues, any type of ethical, legal, social issues, if arises about this project, they have to consult this department, because of only to solve those issues, they themselves design one department so that they can go there and they can consult. So other people, if they are having like so many people will arise some questions, why you are doing like that, why you are finding all these genes and all. To solve those issues, the people should go to this branch and solve their issues in this department. So with this, we'll move on to the goals of Human Genome Project now. So what are the goals? What is their what they want to achieve? The goals are, they are going to identify the total number of genes which is present, nearly it is 20,000 to 25,000. So genes, total genes which is present in our body is nearly it is 20 to 25,000. They are going to identify all those genes. So that is the one goal of this project. And next is to identify, to determine the sequence of the 3 billion sequences. To determine the sequences. How many sequences? 3 million sequences. Sequences means whatever the DNA is having, the nitrogen bases and all those make the sequence. For example, if a strand having the nitrogen bases, nitrogen base like this, okay. So you can call this as a sequence. How many type of sequences are present? Nearly it is 3 million sequences are present. So to find out, to determine all these 3 million sequence, their goal is to find out, to determine the 3 million sequence. And they want to store all these data in the store book. They wanted to store all this data in the storebook. Let me tell you one more thing. I forgot to mention this under the aim. Whatever we are learning now, so I must say, so something is going in your mind. So what is our aim? Why we are studying this concept? Why they put this concept? So we are studying the total constituent of the genes. Do you just think like how many data, how many books they need to write in the to write the base sequences in the typed manner? It requires 3,300 books to type all those base sequences in the book, in the pages. If they wanted to type the base sequences in the pages or in the book, they require 3,300 books. Okay, and each book consists of 1,000 pages, and each page of one book, each page, I am telling you, each book consists of 1,000 pages. Each page must be having 1,000 letters. Now you just calculate yourself how many base pairs are present. Okay, so that is why they call this as a mega project. A mega project. Now, okay, so they wanted to store all the information in a store book or in a stored manner. Now, one more thing I must tell here. Though we are in a goal, they need an improved tools to analyze this data. They needed some improved tools. Tools in sense, it's not like a hammer and all. They need the tools. When I start telling about the biotechnology, they will come to know what the tool indicates. The next is, now as I told you, the LC. So just make it, just write it one more time in the goals. The LC is nothing but ethical, legal and social issues. Okay, so all these things comes under the what? Goals. And they also wanted to transfer these data to the industries. What type of industries? To the health industries, health department, or to the medical department. So many different type of departments or so many different industries, they wanted to transfer their stored data. So that is all about the goals. What is the, I repeat one more time. Why to calculate, why to investigate the total number of genes present in the human body? Let me tell you the application of it so that it will become more interesting concept. So when we come to know the base sequences of our body, it will become easier to find out the mutation, the diseases, wherever the diseases are forming, where the mutation is occurring, we can easily take out that mutated part and reinsert the new and improved base sequence. In total, we can, or the health department can easily solve the diseases and mutation. Not only human beings, not only human genome project, the non-human organisms, the other departments, like not other departments, the same department, they also aimed to sum up, to calculate or to identify the non-human organism genomes. Not only human being, but also some bacteria, some viruses, some animals, birds, flowers, flowering plants, like those non-human organisms, they wanted to study, they wanted to determine the genome of all. Okay, and next is, for example, let me tell you the disease. One disease we learned in the chapter number 5, that is sickle cell anemia. In the sickle cell anemia, you have studied how the sickle cell anemia occurs. Only one base pair get replaced with one more. Only because of the change of that base pair, the amino acid replace the additional amino acid get bind to that sequence. Because of these changes, the mutation occur. That mutation leads to sickle cell anemia. When we come to know the sequence got changed, we can alter that sequence. We can operate that sequence, that base pair, with the proper one, so that we can cure the disease. So not only in the diseases part, we can also improve the food technologies, we can improve the crop production. So number of applications are there. Alright, now coming to methodologies. What is methodology? So methodology in sense how to count the genes. First, what we have to understand here, there are two approaches they made. What are those two approaches? Two approaches. In sense, the DNA, this is having two strands, right? One is going to code with the, one is known as, considered as, one strand is considered as coding strand. To what it is coding? One strand is going to synthesize the mRNA. The strand which is going to synthesize mRNA, that strand is considered as coding strand. One more strand is known as non-coding strand. But here I must replace this coding and non-coding strand with a proper name known as, the coding strand is known as expressed sequence tags. In short, EST. One more strand is known as non-coding strand. And non-coding strand, the name what they have given here is sequence annotation. Okay, sequence annotation. Now, one question arised in this Department of Energy and National Institute of Health. So they were thinking of to count only the coding strand genes, only to identify the coding strand sequences, base sequences. They wanted to leave this non-coding strand because that is not at all in use. It is not at all going to synthesize any of the mRNA strand. So why to simply calculate all the strands and all? It was those are known as two approaches. One to synthesize, to determine only the coding strand, and we should leave this non-coding strand. That is one approach. One more approach came, that is to find out all the base sequences of both the strands. Okay, so out of two approaches, the second approach followed. Why? I'll tell you in the salient features of HGP. In short, HGP. Okay, so the coding strand is known as expressed sequence tags. The non-coding or blind strand or blind approach is known as sequence annotation. So, what they did? They isolate the DNA strand from the cell. Methodologies in sense how they did, how they performed the mechanism, how they performed this procedure. First, they isolate the DNA from the cell. So they wanted to identify all the sequences, right? One by one, they isolated the DNA from the cell. And then after isolation, they cloned the DNA fragments. They cutted the DNA fragments into pieces and cloned the DNA fragments. For this process, for the isolation and for the cloning, they needed the vectors and host. If you are not getting the methodology of HGP, just leave it, no problem. I am going to teach you one more time this concept in the physical class or in this class. I don't know about that. In the chapter number, like in the unit of biotechnology. So need not to worry about that. If I start telling all the procedure of isolation and clone, you'll forget this. That's why I am not going in a detailed way. Okay, just isolation, taking out the DNA and mixing with the other host cell. So the vector in sense, like in simple language, you just understand what is vector. Vectors are just a carrier. Okay, host in sense, so one more organism we are taking, inside that we are making the DNA strand, one more copy of DNA strand only to read out the sequence. Okay. Here the carriers are, the vectors are bacteria and yeast. Vectors are bacteria, artificial chromosome, in short BAC, and yeast artificial chromosome. They used the vectors, the bacterial artificial chromosome, either this or that they prefer to carry the DNA fragments what they have isolated from the human cell. And those vectors with the human DNA, they put it into one host cell. That host cell what they have selected for this process is bacteria or yeast. The most preferred host is yeast. Okay, so the next is, I'm not going in a detail concept, in detail what is that description. I'm not going to tell, I'm not describing all the methodologies and all. Just understand this, taking out the DNA, making it into fragments, and cloning with the vectors and host. Okay, so after the cloning and cloning process, they can read out each and every base pair, each and every sequence of the DNA. The thing is salient features. What are the salient features of HGP? Salient features are: first, they aimed the project. Next, they goaled about the project. The last concept is after cloning and after isolating and all, they came to know the characteristic feature of the sequences and genes. So what are the actual sequences and all, actual salient features and all, that we'll understand here. The salient feature, the first salient feature is there are about 3164.7 million base pair. So I am going to teach you about the salient features of HGP. Salient features are as follows. Remember, my dear students, the most expected question is salient features, and for 5 mark, and the goals and applications, maybe they'll ask you to write for 2 or 3 mark, but don't forget to learn this salient feature. The first salient feature is there are about 3164.7 base pairs present in a human genome, Okay. The next is average gene contains 3000 bases. 3000 bases. It means what? Average genes means what? Not a short gene or not a long gene. An average out of all the length of gene. If we take an average length of the gene, an average means, for example, this much of gene, the maximum number of gene length will be this much. Okay, just imagine, suppose this average gene consists of 3000 base. Now I have written only 20 to 30, 25 bases. So just you have to imagine, in one gene, average gene, 3000 bases will be present. Okay. Now, total number of gene is, so the longest gene is considered as dystrophin, having the base pair 2.4 million. It means what? Dystrophin is a gene having 2.4 million base pair. Now you just imagine how long the gene is. And one more thing I must say here, out of all the discovered gene, out of all the known gene, only 50% of gene functions are analyzed. The other 50% gene functions are unknown. So that is why they chooses the second approach. We don't know which strand is going to synthesize the protein, not the, what is that, not strand, only the coding strand will synthesize the protein, which they doesn't know which DNA fragment is having different function in the human body. Only to make that analyze, only to make that determine, to make that clear, they selected the second approach. Second approach was to find out all the DNA base sequences. All the DNA base sequences means both the two strands sequences. After that, they cannot discover the 50% gene function. The other 50% gene function is known, but the 50% of the gene function is unknown. Okay. And the next is the chromosome 1 has the maximum genes like 2968 genes it contains. And chromosome Y, this is the maximum number of genes present in a chromosome, and chromosome Y has least genes in it. Genes are mentioning here, not the base pair. Here I have mentioned 2.4 million base pair. Here I am mentioning 2968 genes. In one gene, an average gene consists of 3000 base. Don't get confused with all these. Chromosome Y consists of least number of genes. Okay. The next is the repeated sequences makes up the large portion. Repeated sequence makes up large portion of gene. Okay. Like a repeated sequence, for example, if I am having a strand here, number of bases in it, if we write the sequences blindly, if we write like A pairs with T, G, C, C, G, A, T, T, A, T, A, G, C, C, G, like that number of bases. The repeated sequences. So this is one strand. Okay, one more strand, if we take this will be having, let me write one more thing here, C, C, C. If I keep on writing only C's, this will become a repeated sequences. This type of sequences makes a large portion. One more thing, that is the sixth character I must tell here. Just suppose this is one strand having different bases. One more strand will be having all the base pairs similar to this strand, but only one base pair will get differ. Only one base pair will get differ in this. For example, here I wrote T, A, T, T, A. Leaving this base pair, one base pair, single nucleotide bases, all the other bases are similar, if suppose. So that type of nucleotides or nucleotide sequences are known as single nucleotide polymorphism. In short, SNPs. SNPs having one difference in the nucleotide bases out of number of strands. That concept is known as single nucleotide polymorphism. So, these are all the different salient features of a human genome project. After reading all the bases, they came to the characters of genes. When they studied all those, they find out number of characters like this. Few of those they have given in your syllabus. Just learn it. If you have any doubt, just ask me.